Good afternoon, everyone. I'll be presenting on the GC used in aviation fuel testing. The aviation industry is determined to develop sustainable solutions to produce cost-competitive, high-yield jet fuel with minimal environmental impact. It is common for some of the government to mandate the use of renewable components. The introductions of biodiesel into many countries has created a new threat to jet fuel quality. Thus, we will require the solution for this analysis to ensure the quality of the fuel is unaffected by the missing proportions of renewable components. There are three types of traditional testing methods, which are GC for simulated distillation, SIMDIS, GC for film content, and GC by GC for the compositional analysis. Simulated distillation. We are using D2AA7 in this application. This is a test method used to determine the boiling point distribution of petroleum products by GC method. It can be used as an alternative to D86, which is the traditional distillation method using the gas burners, condenser, and the distillation setup. According to D1655 that I extract it, you can see that there is one statement mentioned that D2A7 gives true boiling point distributions, whereas for D86 is a low efficiency distillation method. With ASTM D2A7 data, it can be converted to D86 data using the correlation feature with our SIMDIS Excellence software. Besides, it also creates significant benefits for analysts. First of all, PAC proprietary TPI, temperature programmable inlet, can ensure sample integrity. The sample is injected at low temperature and with the temperature ramping in the inlet to avoid sample loss on the lighter ends that will lead to sample discrimination. Second, it is safe. It eliminates the potential fire hazards. And lastly, it improves throughput twice as fast as D86 as it is a fully automated solution, can save up to 75% reductions in operator time. Okay, let's look at the actual sample run on SIMDIS, the actual sample. As you can see, the true boiling point data for D2887 and also the correlation data for D86 can be obtained using the same software. So you might have questions, how reliable the correlation data is. So here comes to the next slide to explain to you about the correlations of SIMDIS to D86. The historical data show an excellent correlation of SIMDIS to D86. So the graph and tables show some result. As you can see in the box that I highlighted in yellow here, this is the correlation data, and this is the D86 data, the actual run on D86. The delta between the correlated and D86 is less than 2. This system can be used to run D86 group 1 and 2 because they are using same configurations and same column use. It shows highly accurate results for gasoline, for this FCC naphtha and reformer feed. As you can see from the correlation data here, they are nearly overlap on two lines together. Thus, PAC SIMDIS is a good solution for SAF boiling point determinations, and it also shows excellent correlation data with D86 results. Next will be FAME analysis in aviation turbine fuel. FAME in aviation turbine fuel, we are referring to IP599 method. This is the test method to analyze FAME traces in jet fuel using GC. As per D1655 standard specification for aviation turbine fuel, IP599 is allowed to be used as an alternative for IP585, which is a GCMS method. So this is not required to be analyzed every batch unless there is risk of contamination that would exceed the limit. Our FAME in aviation turbine fuel analyzer offers the best return on ROI, best return on investment, and best precision and accuracy due to the FID detector use. In this analyzer, we will be using hard cut and non-query refocusing technique. After first dimension separation, the specified FAME species are transferred towards a highly polar second dimension column by doing a hard cut. Before the final separation is performed, the FAME species are trapped by the temperature-based refocusing trap. So let's look at the flow configurations on the next slide. As you can see from the flow diagram here, this is the refocus trap. This refocus trap will make use of air vortex to create cooling effect to bring down the temperature to around 25 degrees Celsius to trap the film content. So cooling of this refocus trap is done by using compressed air with a minimum capacity of 30 liters per minute. And as you can see from the first graph, the first dimension column gas is the cold eluted peaks. So it has to be re-injected into the second dimension column to improve the separation so that all of them are thoroughly separated. And you may also find that C17 peak is disappeared in the second dimension column because it is not the target of interest and it has been cut off so that you will not see it in the second column. So in short, PAC film in jet analyzer is the best solutions for measuring film in jet due to the great precision and cost effective than other detector. Next is the GC by GC application for the compositional analysis. This is the PAC effort to introduce this method into the SDM under D8396. D8396 is a test method to quantitatively determine the paraffins, isoparaffin, naphthenes, and aromatic up to 2-ring plus. The main principle of this method is a portion of the compound is separated on the first dimension column and then sent to the second dimension column for further separations. All sample analytes will undergo 2 different separation mechanisms. So for example, first will be by polarity, second will be by boiling point. One thing to emphasize here is this flow modulator. These flow modulations designed and used by AC does not require cryo setup. For information, cryogenic modulation will be relying on the cryolic fluids or cryoliquids for the focusing functions. It is expensive and impractical for field use. So this flow differential is used to inject the modulated peak into the second columns here. So you can refer to these small peaks. Initially, what cannot be separated in the first column will be able to separate in the second column with the better separations and higher sensitivity. So if there are 2 cold elutions compound in this single peak, these slides of cold elutant peak will be injected quickly into another column with different selectivity. The GC by GC modulation basics. These diagrams illustrate the basics of GC by GC modulation. In diagram 1, this is a broad peak produced on the first column that contains 3 cold elutant peaks, the red color one, the yellow color, and the green color peaks. So they are cold eluted as the big peak shown in blue. So in diagram 2, the first column peak is modulated, and each of these 9 modulation segments are transferred to a second column with different polarity. And lastly, the 3 cold eluting peaks are now separated, and this is what the raw 2-dimension data looks like. These are the real conventional jet fuel and SAF data. As you can see from the conventional jet fuel data, there is a small amount of aromatic content. So usually it is minimum 8% to maximum 25% to prevent fuel leakage and ensure seal tight for the engine and aircraft. Whereas the rest of the 3 SAF sample doesn't have or have a very insignificant amount of aromatic content in their SAF samples. So for this data, the data acquisition is done by OpenLab CDS, but the data is too messy that you will require additional software for data processing called GC Image. So this data is actually from GC Image. In short, GC by GC is an excellent solution for individual hydrocarbon analysis in percent level. A quick summary. So we have discussed about 3 GC applications: SIMDIS, FAME in Aviation Turbine Field Analyzer, and lastly is the GC by GC application. Each of these analyzer is factory tested and comes with a startup kit to allow for fast commissioning. If you have any queries, please contact PAC for further information on the solutions for GC analysis. Thank you.